Hello, my name is Joshua Mugabe. I welcome you to Immune System. What is the function of the immune system? The immune system performs specific defense against agents, those are the antigens, that the foreign and harmful substances to the body. Exogenous antigens, they are often in contact with the skin or entering the airways, the digestive tube, the genital orifice, and the mucosa. They can also penetrate the circulation directly through the wounds. What are the two groups of defense mechanism the body uses against foreign and harmful agents? What is the difference between them? The body has many defense mechanisms against foreign pathogen agents. These mechanisms are divided into two groups. We have the specific mechanism and the unspecific mechanism. The specific mechanism are part of the immune system and comprehend the humoral immune response and cellular immune response that respectively produce antibodies and defense cells against specific antigen. The unspecific mechanism fights in general manner any type of antigen. They do not have specificity, and in them cells of defense means are included, like for example the skin barrier against foreign agents, mucosa and ciliated epithelium of the airways. There is inflammation, the inflammatory response, the action of unspecific proteins and defense cells, the interphons and the macrophons. What is inflammation? Inflammation is the initial response of the unspecific defense system versus aggression against the body. The aggression may be caused by infectious parasites, chemical contamination, trauma, physical agents like heat, fire, autoimmunity, and many others. During inflammation, a cells of unspecific leukocytes present in the circulation are attracted to the injury site in an attempt to destroy harmful agents and isolate the affected region of the tissue. How does inflammation mechanism work? When some tissues injury occur, the histamine and other vasoactive substances called mediator of inflammation are released. They cause vasodilation and blood flows in the affected site increases. Granulocytes, leukocytes present in the blood are attracted to the site of injury by substances known as chemotactic factors, also released by the injured tissues, and by the active granulocyte in the area. The granulocyte exits the capillaries by their pedicis, i.e. that is using pseudopods. Macrophages present in the region are activated too. The cells flood the extracellular space of the affected area, trying to kill and eliminate harmful agents, to prevent tissue necrosis, and to isolate the damaged tissues. What is PAS? PAS is a residue of the inflammatory reaction. It contains a mixture of fragments of dead leukocytes, infectious agents, and tissues. What is the association between inflammation and fever? In the tissue region where inflammation occurs, bacteriotoxin, cytokinins, prostaglandins, interleukins, endothelins are released. And these substances gain circulation and reach the central nervous system, which then commands an increase in the body temperature. What type of defense cells do bacteria attract and cause multiply during the inflammation process? What is the name given to the waste material produced by the inflammation triggered by bacteria infection? The main leukocytes that generally multiply and participate in the inflammation reaction against the bacterial infections are the neutrophils. And this type of inflammation, the blood level of these cells are increased, a clinical condition known as neutrophilia. The bacteria inflammation fragment and the dead bacteria, dead neutrophils, and tissue all form what we call PAS. Of which type of defense cell do worm infections stimulate the multiplication? The main leukocytes generally multiply and participate in the defense against worm infection are the eosinophils. And this type of inflammation, the blood level of these cells are increased, a clinical condition known as eosinophilia. Eosinophils are also increased in allergic conditions. Of what type of defense cell do viral infections stimulate the multiplication? The main leukocytes that generally multiply and participate against viral infection are the lymphocytes. In this type of inflammation, the blood level of these cells are increased, a clinical condition known as lymphocytosis. What defense mechanism that begins the work of when inflammation fails to stop an infection? If the inflammatory attack is not enough to halve the infectious process, the body still relies on a specific defense, the immune response proper, or humoral or cellular, performed by lymphocytes. What is the difference between humoral-specific immune response and cellular-specific immune response? Humoral-specific immune response is a defense system by means of antibodies, defense proteins secreted by lymphocytes that attack foreign agents with high specificity. Whereas cellular-specific immune response is the defense system by means of specific lymphocyte cells that directly attack the foreign cells and agents. What is an antigen? Antigen are substances, particle, infectious agent recognized as foreign to the body. The contact of antigen with the body promotes defense reaction against the antigen, and specific, or both. What are the cells responsible for the production of antibodies? The cell that produce antibodies, that is, the cells of the humoral immune response system, are the B lymphocytes, or what we call the B cells. What is immunoglobulin? Immunoglobulin is an alternative name given to the antibody. Immunoglobulin are complex protein containing invariable portion and variable portions made of 4 polypeptide chains. The variable portion of each immunoglobulin is responsible for high specificity of the antigen-antibody bond. How do antibodies work to neutralize antigen? The antibodies or immunoglobulin act to facilitate the destruction of antigens. They attract phagocytotic lymphocytes. They trigger the attack of specific defense molecules, that is activation of complement system, and they directly neutralize the toxicity of some antigens. How can an organism that once underwent contact with the antigen be immunized against future infection of the same agent? The phenomenon called immune memory, when the antigen make contact the first time with the cells of the humoral immune system, B lymphocytes that are producers of specific immunoglobulin against the antigen multiply in the days, synthesize their antibodies. This is called primary response. Some of these specific B lymphocytes remain in circulation for a long time, sometimes during the entire life of the individual, and they become the memory cell of the immune system. When the body is exposed to the feature of the same antigen, the production of antibody will be faster and more intense, since the immune system already prepared to react against the antigen. This is called the secondary response. How can the immune memory lead to the efficacy of vaccine and also produce allergies? Vaccines are controlled inoculation of fragments of infectious agents, of inactive infectious agents to induce primary immune response, the formation of specific memory B lymphocyte against the antigen. Therefore, the organism produces immunoglobulins and becomes prepared to destroy antigens when exposed to new infections by these agents. In allergies, the humoral immune response is sensitized, makes the antibody and specific memory B lymphocytes against some common environmental substances wrongly recognized as antigen. For example, pollen-derived substances, dusty particle compound present in the foods or in medicine and so on, may be recognized as antigen, triggering the primary response and creating immune memory against them that then becomes the cause of allergy. The more the individual is exposed to these substances, the more intense the immune reaction. The 1GE antibodies that cause allergy bind to receptors of leukocytes, called mastocytes, whose cytoplasm is full of histamine granules. The antibody-mastocyte bond causes the cell to release the agent amount of histamine in the circulation, stimulating inflammation, generating allergic symptoms and signs. For this reason, allergy is treated with antihistamines, drugs that block histamine reaction. It is a beta allergic reaction, for example, hypersensitivity to some medicine like penicillin, sulfurs, may cause anaphylactic shocks, severe clinical condition that sometimes can lead to death. How different are the actions of antibodies against bacteria and against virus? Why is the cellular immune response activated in case of chronic viral infection? Antibodies of the humoral immune system act against extracellular agents like toxin or bacteria, but they are not active in the intracellular space. They cannot fight viruses efficiently. In the case of viral infection and also cancerous and precancerous cells, the immune attack is made by cellular immune system, mediated by T and NK, natural killers, lymphocytes, that destroy specific cells and viruses. How does the cellular immune response take place? The lymphocytes that participate in cellular immune response are the T lymphocytes. T lymphocytes differentiated into three types: cytotoxic T lymphocytes, cytotoxic T cells, helper T lymphocytes, helper cells, and suppressor T lymphocytes. Cytotic cells are the effectors of the system. That is, they directly attack other cells recognized as foreign. For example, fungi cells infected by viruses, neoplastic cells, graft cells, and many others. The helper cells and the suppressor T lymphocyte acts as regulators of the system, releasing substances that respectively stimulate and inhibit the immune action of the T and B lymphocytes. After primary immune response, memory T lymphocytes also remain in the circulation to provide faster and more effective reaction in case of the future infections. What are the antigen-presenting cells in the immune system? The antigen-presenting cell in the immune system, also known as APC cells, are cells that do phagocytosis and digestion of foreign microorganisms, and later expose the antigen derived from these microorganisms in the outer side of their plasma membrane. These processed antigens are then recognized by the lymphocytes that activate the immune response. Several types of cells like the macrophage can act as antigen-presenting cells. What are passive and active immunization? According to the duration of the protection, how does this type of immunization differ? Active immunization is that in which the antigen penetrates the body, triggering primary immune response and the production of memory lymphocytes and antibodies that provide faster and more effective immune defense in future infection by the same antigen. Passive immunization is that in which the immunoglobulin against the antigen are inoculated in the body to provide protection in case of the body become infected by the antigen. Active immunization tends to be longer lasting than passive immunization, since active type as well as antibodies, specific memory lymphocytes remain in circulation. In passive immunization, the duration of protection is that of the duration of the antibodies in the circulation. Why is maternal milk important in the immune protection of the baby? Besides being nutritionally important, maternal milk participates in the defense of the baby against infectious agents. Soon after delivery, the mother produces more fluid milk called colostrum that is rich in immunoglobulin, that's the antibodies. The antibodies are not absorbed by the baby's circulation, but they cover the internal surface of the baby's bowel, thus attacking possible antigen and making more difficult for the proliferation of the pathogen bacteria within the organ. How are the antivenom produced? Why are antivenoms an example of passive immunization. Antivenoms are obtained in the following process. The venom, which is the antigen, is inoculated into another animal, for example, the horse, and this animal makes specific antibody against the antigen. Blood from this animal is collected and purified to get the antibodies. This antibody-containing material is the antivenom. When human beings are infected with the antigen of a specific antivenom is given to him or her, and the action against the antigen occurs. Antivenoms may also be administered as preventative measure, since it is basically made of specific immunoglobulin against some antigen. The process is an example of passive immunization. What is the difference between homologous and heterologous immunoglobulins? Homologous immunoglobulin in the human from the same species, immunoglobulin, in the case of inoculation in animal, as in veterinary produces homologous immunoglobulin, is that from the blood of the animal of the same species of the animal undergoing treatment. Heterologous immunoglobulin is that obtained from animal of different species from the individual into which it will be inoculated. The homologous immunoglobulin is safer since it is collected from being of the same species of the individual in which it will be inoculated, and thus the risk of antibodies to be recognized as foreign to trigger immune response is lower. Heterogeneous immunoglobulin are more prone to being destroyed by the own antibodies of the individual. What are the natural active immunization and artificial active immunization? Natural active immunization is that in which the previous natural infection induces a primary human immune response. Specific memory cells are produced and the individual become immune to the new infection with the antigen. This is what happens in the disease that affect people only once in life, like for example the mumps and chicken pox. Artificial active immunization is that in which the primary immune response is caused by inoculation into the individual of specially prepared antigen, in this case with the vaccine. Why are vaccines made of their own disease agent or the fragment of it? The goal of vaccine is to artificially induce a specific primary immune response and the consequent formation of antibodies and memory cells concerning a given infection or disease in order to immunize the individual against infection by the pathogenic agent in the future. Since each antibody does not act against a variety of antigen, but instead it acts only against a specific antigen, it is necessary for the immune system to make contact with some way with the antigen against which the immunization is wanted. The reconnaissance of specific molecular portion of each antigen cause the immune response to produce specific variable portion of the immunoglobulin to attack the antigen. Therefore, to induce the active immunization, it is necessary to inoculate into the body small parts of the infectious agent or the agent entirely, which is dead or inactivated. What are the types of antigenic agents that may constitute the vaccines? Vaccine can be constituted by dead agents of the disease, of inactivated agents of the disease, of inactivated toxin, or of fragments of the infectious agents. Examples of vaccine and their type of antigen agents. BCG, inactivated tuberculosis bacilli. Anti-tetanic vaccine, inactivated toxin. Anti-diphtheric, inactivated toxin. Anti-polio, dead poliovirus. Anti-poliosabin, attenuated inactivated poliovirus. Why doesn't long-lasting vaccine against the common cold exist yet? Viruses that present high mutation rate, like viruses that cause the common cold, escape easily from the action of vaccine against them. After the primary human response, natural or artificially induced against the virus, the next season of infection, new mutant-resistant strains appear, and the protection obtained with the immune response of the last season is lost. One could say that the high mutation rate is a form of immunization formed by these viruses. Why are vaccines used in the prevention but not in the treatment of infection? Why can antivenom xylem be used in prevention and treatment? Vaccines are not used in the treatment of infections because they depend on primary immune response that takes about a week to occur, is not so intense and effective. Antivenom serum, however, are inoculated into the circulation and used as immediate treatment because they are made of great amount of immunoglobulin antibodies, which is potent against the respective specific venom. What is DNA vaccine? DNA vaccination is a vaccination technology based on genetic engineering. In DNA vaccination, recombinant plasmid, the vector containing the gene of a specific antigen, that is part of the given pathogenic agent is inserted into cells of individuals to be immunized. These cells begin to produce the antigen to trigger the primary immune response, and theoretically the individual becomes immunized against the antigen. What is the name given to the condition in which the only human system of the individual is the agent of the disease? What are the examples of these conditions? Disease caused by the action of the own immune system of the individuals are called autoimmune disease. The autoimmune disease appear when the immune system makes antibodies or defense cells that attack cells, tissues, and organs of own body. The attacked cells and tissues are wrongly recognized as antigen by the immune system. Rheumatoid arthritis, lupus, scleroderma, vitiligo, pemphigus, type 1 diabetes mellitus, Crohn's disease, chronic inflammation of the gut, myasthenia gravis, Graves' disease, Hashimoto's disease, and many others are all examples of autoimmune diseases. Thank you very much for participating in immune system. Please do like, share, and subscribe.